Hello everybody, my name is Kirtana and I will be presenting a paper titled Oligodendrocyte Ablation Triggers Central Pain Independently of Innate or Adaptive Immune Responses in Mice. According to the CDC website, a 2019 survey concluded that 20.4% of adults suffer from chronic pain and 7.4% have low quality of life because of it. Although the mechanisms underlying chronic pain remain elusive, gliopathy, which describes dysfunctional glial state, has been postulated to be a key contributor to neuropathic pain. Studies in chronic pain have not addressed the role of oligodendrocytes yet, but their role in supporting axonal functions independent of myelination has been well established. This leads to the question of whether there are functions or pathologies of oligodendrocytes linked with pain that are not addressed yet. The researchers have used a plethora of techniques, but I will be discussing only those employed to get the results which we will be seeing in a bit. Transgenic mice expressing diphtheria toxin receptor, or DTR, under the expression of MOG, which is short for oligodendrocyte-specific myelin oligodendrocyte glycoprotein, were engineered using the Krelok system. Intraperitoneal injection of diphtheria toxin, or DTX, was administered for 3 consecutive days. Behavioral tests were used to assess sensory dysfunction. Time to withdraw paw was measured to graded force or graded Monfrey hairs and temperature control plates in mechanical and thermal sensitivity tests, respectively. Motor coordination was measured using rotor rod, and time spent on the rotating rod was considered. Immunohistochemistry and cell counting was used to probe and detect specific proteins and cells. Time points of 6, 16, and 24 days post-DTX injections were considered for most experiments in the study. DTX-mediated ablation was validated by reduction in cells expressing ASPA and APC proteins. These proteins are expressed specifically in the somata of oligodendrocytes. The images here show a sharp decline in oligodendrocytes in the spinal cord, and the quantification suggests significant decline in the transgenic mice compared to the control group, which suggests a successful ablation of oligodendrocytes. This led to significant decline in sensory dysfunction as observed here. We can see a sharp decline in the threshold to mechanical and non-noxious cold stimuli setting in at about 5 to 6 days post-DTS injections in transgenic mice compared to the control group. We can also see a drop in the motor coordination 20 days post-DTS injection. It progresses for the next 10 days significantly. These results show that ablation of oligodendrocytes affects nociception in adult mice. Injury in spinal cord is associated with activation of microglia and astrocytes, which are measured by probing for CD68 and GFAP. These are markers specific for phagocytic microglia and astrocytes, respectively. CD68 immune reactivity and GFAP positive astrocytes are observed only at day 24 following DTS injections in the experimental group compared to the control group. This shows a delay in the transformation of microglia and immunoreactivity of astrocytes compared to onset of mechanical hypersensitivity and cold allodynia at 5 or 6 days, which we previously saw post-DTS injections. So the trend observed with innate immunity sort of carries over to the adaptive immune response as well. In this slide, we see that T cell and B cell mediated response shows no significant difference between the two groups. The adaptive immunity did not kick in during the time points considered, suggesting a delay in the immune response. From the set of experiments presented here, the key points are that oligodendrocyte ablation leads to nociception in response to mechanical and non-noxious cold stimuli. Onset of cold allodynia and mechanical hypersensitivity was not associated with microglial reaction, astrocyte transformation, or T cell responses. This suggests that widespread loss of oligodendrocyte causes nociceptive disbalance in a mechanism independent of innate or adaptive immunity. Hence, in the context of pain treatments, targeting oligodendrocyte function may be of value, and teasing out the mechanisms of how loss of oligodendrocyte leads to nociception can also provide new targets for therapy. Thank you all for watching.